Okay, I'm going to go ahead and get started. We are going to pick up with the GWAS slides from last week, Thursday. All right, so this is back in Module 7, Thursday. I am going to be going over this PowerPoint, which we only barely got started last Thursday. Somebody summarize where we are in genome variant analysis. What have you done so far? Just want to make sure we're on the same page before we start doing the next thing. Yeah, so two things there. What do they mean, and are they real? Are they a real variant? That's kind of where we're at, right? Like what we've done so far. We did quality control, we did mapping, and then we did variant calling. So in principle, after we've trimmed the reads, mapped them, and called variants, what the picture that you submitted yesterday showed, I would have to rerun my Colab notebook. Actually, maybe I wouldn't. It might already be generated. Nope. Yeah, so hopefully you remember what your picture looked like, right? It's got a circle with a bunch of black, it looks like they're just crosshatches. What were those black lines all the way around? Did anybody read the code or try to figure out what it was doing? I mean, it's commented so that if you looked at the comments, you may know. It was the genome, but all of those black sort of tick marks around the outside of the genome. No? Everybody's looks the same, so it won't matter if I pull. Right? So this is a picture that everybody got. So what is that outside ring showing? It is the genome, but then those black tick marks are demarcating the genes. Okay? So those are the coding regions. And then the blue and the red tick marks, what are those? Those are the variants. What's the color difference mean?
Red is coding, blue is non-coding.
Yeah, good. So the red ones, if you zoomed in, and you can't do it on the static image, but the Plotly thing that the Colab notebook actually produced allowed you to zoom in so that you could see which gene those hit. Okay, so this is where we've gotten to so far in our genome variant analysis pipeline. We have identified variants, and I guess maybe you could argue that identifying which ones are coding and which ones are non-coding gives you a little bit of an idea about what they might do, whether they're functional or not. But we really don't know what any of these things do yet, whether they have a phenotype at all. And so that's what the GWAS is all about. So genome-wide association study. I talked about the workflow, and then slides. But essentially, this is what we are going to try to do. The data that we're going to run our Colab notebook is the same kind of phenotype data where it's binary. You're either a case or a control. You have the trait or you don't. And then every row here is an individual, and every column is a SNP or a SNV. So, a red or a blue tick mark on that circle that we just showed. And then we are going to use logistic regression to say, is there an association between whether you're a case or a control? Are the SNPs across this individual identifying whether that person should belong to the case group or the control group? So graphically, if you look at what's going on in that table, SNP number 1, right, if you scan across all these individuals, there's really no association whether they have an A or a T and whether they're a case or a control. If you slide over to number 4, which has a high negative log P, you can see it's almost a perfect match. If you have the C, you're a case. If you have the T, you're a control. So that would be a thing that's highly associated with whatever the phenotype is here, okay? And then SNP number 5, maybe a little less, 6 and 7, not associated. If you look at that across the genome, right? So instead of having 7 SNPs, now we've got millions of them. You get a Manhattan plot kind of picture. And then we're going to talk about where you draw that dotted line. So what are you going to call an association? We'll talk about that toward the end. And I think this might be the last slide that I got to Thursday. The QQ plots. So one of the things we're going to look at in the Colab notebook are the outputs for QQ plotting. And the way to think about this is, if you had purely random association between the SNPs, like you just drew alleles for every SNP, no, disregarding the phenotype, you would get a distribution of associations. Okay, that's how frequency statistics work. You're going to have most of them that, because it's random, they're completely unassociated with the phenotype. But occasionally, random pulling is going to give you a perfect association with the phenotype, okay? And so these QQ plots are showing usually it's the observed on the Y and the expected on the X. So, right, most of them are going to be very low, where you have these are typically plotted in negative log P dimensions, where very low means not associated at all. And so if you put them in rank order from the lowest, basically not associated, to the highest, then you would have a expectation of all of those SNPs falling on the diagonal if there are really no associations, right? Because that's like the random pulling alleles from the bag, okay? So the way that you see on a QQ plot that there's actually some signal, there are some things that are associated, is when the actual p-values for the association start to deviate from that random expectation. So you can see in all of these disease examples, you get some p-values in the observed set that are higher than what you would expect based on a random distribution, okay? So if everything was random, you wouldn't have these black dots sort of shooting up on the observed axis, okay? And so that gives you an idea that there are some SNPs in these data sets that you should expect to find above whatever significance threshold. And if you look at the Manhattan plots for these diseases, right, bipolar, you might argue that there's not very much that's strongly associated because the observed values mostly fall right on that expected line for the distribution of test statistics. And if you look at the bipolar SNP Manhattan plot, you do see there are barely any. You'd have to really zoom in to see there are a couple green dots that are barely above the background. But then for some of them, right, the Crohn's disease, coronary artery, where you get really strong peaks, there is a strong association in some regions. And so these are just kind of zoomed in. Why do you see these clusters of significant SNPs? We'll get back to this question in a minute, but just curious about your intuition.
I feel like certain genes have a stronger control over whether a patient would have a disease or not.
Yep. Could be real, like you've got more than one mutation in that gene. What's another explanation? This is population genetics world, so I just didn't know if you have any intuition about it. But linkage, remember, back to your genetics, if there's any population structure, and so the fact that you have a causative variant, for example, and there's population structure, means that you also have other SNPs that are just linked to that one. If there's population structure, different groups are going to have different linkage patterns, and so you're going to get these clusters around the significant one that are just because of your population structure. Okay. So we'll come back to this, you know, how do you tell what's causative and what might just be linked? Odds ratio. So, you know, especially if you're in a clinical setting, telling a patient who had genetic testing that the p-value on their GWAS was, you know, 0.005, you know, what does that mean for my probability or my susceptibility to developing this condition? Not very useful. And so in the clinical situation, you often see odds ratios, and this translates into like how many fold increase do you have because you have a certain genotype. So this table is just showing you, for example, if you want to know the odds ratio for individuals that have the GG phenotype versus the CC, I said phenotype, I meant genotype, right? Homozygous G versus homozygous C, right? It's just this cross multiplication and division. Putting numbers on that, right? So you would do sampling where taste and not taste. This is taken from a taste receptor gene example. I forget if this is the cilantro or the broccoli one, but right. If your GG, 12 homozygous Gs can taste it, only 8 can't. And then if you look at the other genotype that we want to compare, 58 homozygous Cs can't taste it, 3 can. And so, right, you're doing that cross multiplication, 12 times 58 divided by 8 times 3, and that's a huge difference, right? So your odds ratio of being able to taste this thing is way stronger if you have the GG phenotype. Okay, so let's actually play with some data. There's a link there to a Colab notebook. Oops, not that one. So last Thursday's lecture, the Colab links, this GWAS tutorial button, so you should open that. I don't know why it wants to. Okay, and we'll walk through this the first time together. This is going to, in our first example, going to simulate some data, and then I'll have you work on some chromosome 22 human data. But first step is to run this cell that installs the dependencies. It's Sci-VCF2 is something that'll help us convert a VCF to a genotype matrix, because right, this, where's my PowerPoint? We need something that looks a little bit more like this, not like your GCF file, where it has rows for SNPs. Okay. And then Pandas is a data frame manager, so we can treat things like a big table. And stat models is what's going to let us do the logistic regression. So first time through, we're not going to run this cell because that will ask you to upload data. We'll do that the next time around. The next cell, we'll check and see if you've uploaded a VCF, and if not, it's going to simulate SNP data for 100 individuals and 200 SNPs on each individual. So you should see no VCF found, forcing synthetic. So, and then you should also see the simulated .vcf pop up in your, and depending on how often you actually looked at your VCF, or if you just took it out of Slurm and stuck it into Colab, this is what a VCF file looks like. So that's what just got generated by that cell. And so that's what I mean by here we've got rows of SNPs, and we need rows of individuals with the SNPs in columns to do our associations. So that's the next cell. Read that VCF file and turn it into a genotype matrix. Everybody following so far? It's working. So next step is to load phenotypes. We don't actually have phenotypes for this one, so we'll let, if you hit cancel, it's going to simulate. So it's just going to generate case control data across all, I don't know if it does it randomly. Actually, I think it does. But oh, so, yeah, I told it to do a slight genetic effect. You guys have the nicest ringtones. Okay, so next we've got our genotypes and our phenotypes. So essentially we've got that table, and now we're going to run the logistic regression on all of those SNPs. So this is going to run a bunch of logistic regressions, and it's just showing us the top of that table. And here it's just giving us the SNP number, the chromosome that it belongs to, the start position, that's the base pair, the p-value of the logistic regression, and then beta. This is again, this is an estimate of effect size. Think of it as like the slope in a logistic regression. So it tells you how different, or what is the strength of the association between that SNP and the phenotype that got simulated. All right, and then next cell, we've got the associations done, and now this just generates an output that creates odds ratios and confidence intervals. So you can open that file. It's kind of messy here. You could download it and open it in Excel if you want. Really, that cell is mainly just so that you can see effect sizes, and we may go back and look at that when we run the other data set in a few minutes. Next, we'll generate the Manhattan plot. And just out of curiosity, does yours look anything like that? Yeah. So what does that mean? You've done your first genome-wide association study. What does it mean? And granted, it's all simulated data, so you don't really, the phenotype is arbitrary here, but what would be your interpretation? Okay, biological question is there an association between the genotypes and the phenotypes? I see some heads shaking. No association, right? So none of those dots go above the dotted line. Now, I said we'd get back to that dotted line. What does that dotted line mean? Actually, okay. Okay, so going back to the PowerPoint, hopefully that collab, I don't know how much just got cut off, but hopefully that ran okay for you. If you look at the association table, which we opened a minute ago, those are the columns that are there, again, with the odds ratio and lower bounds of those based on the logistic regression associations. Already hinted at this. So this is beyond just, whether it's statistically significant or not, but if you find an association and it's between marker locus, which is one of the SNPs that you found an association, and the disease or whatever the phenotype is, there are multiple interpretations of that. One, it could be causative. Ideally, that's what you're looking for. Alternatively, it could just be correlated with it due to linkage disequilibrium. That's what I was talking about with population structure. You can also have other ways that population substructure, I guess these are really tied together and whether there's population substructure and linkage disequilibrium. But the reason they're different is that some populations, so in the human population, for example, all non-Africans have much higher linkage disequilibrium than all African populations. And that has to do with the age of the populations. So this is important in human genetics because the probability of something being associated just because it's linked is higher in, say, European populations, just because there's been less time for recombination since the bottleneck of those populations. Everyone outside of Africa is more closely related to each other than like two subpopulations within Africa. A whole, like, we could do a whole lesson. I've got an example here on, actually, let me just skip to it. This is what I'm talking about. If you look at, this is human population structure, right? So these are the African populations down here, some South and East Asian populations there that come off here. But right, this really deep split, it's Ethiopia, East Africa, and then there's Central Asia and South Asia. And then this is what I mean by everybody else is more closely related to each other than, say, two populations from that group and that group. And so that creates issues when you're trying to do genome-wide association studies in humans, because there's a tremendous amount of linkage disequilibrium in these populations where, you know, you've got huge chunks of the chromosome where all the SNPs tell you the same thing because they're not independent of each other. They haven't recombined. So let's look at a sort of, actually, population stratification just as a definition leads to false positives, right? So all of those linked markers are potentially telling you there's an association when they're really only linked to the thing that is causing it. So real case or real example, cartoon example of how this can cause you to go wrong. So if you went out and sampled and you didn't know that there was population substructure in your sample, you treat all of these as one big pool, which is what we kind of just did in our 100 individuals with 200 SNPs. And you do your genome-wide association studies, and you get the case control information. You can run this through the GWAS, and it'll tell you there's an odds ratio of 1.2 and a p-value of 10 to the minus 8. And you say there is or is not a correlation there. So an association with that. And so let's talk about the, like, that's problematic because you've got subpopulations with different allele frequencies. So underneath the blue and the red, you can see that the A allele is only at frequency 0.2 in that population, and the frequency of A in this population is 0.5, and the phenotype, in this case diabetes, is much lower in this population than it is in this population. If you analyze this as one big population, you don't know that there's this sort of confounding effect where the two populations have different frequencies of the A allele and different frequencies of diabetes. This is probably going to give you an association with the A, even though that doesn't necessarily mean that allele is associated with it. It's just due to the population substructure, right? And so what would you want to do with that data? Like if you really want to know if there's an association, how would you fix that? That would tell you, yeah. So you could do that if you know how to split the individuals, or you could do some kind of permutation to see if it can split it. We're going to talk about, yeah, when we get to machine learning, we're going to talk about ways that you can discover that structure. But given that you know this is happening now, how would you do the analysis to see if there's actually an association? Oscar? Yeah, so correct for the different allele frequencies or different phenotypes. If you know which individuals belong to which population, you can just do the GWAS on that population, right? So if we did our GWAS separately for the blue group and the red group, which is what this slide does, right? Whoops. Man. Then what you end up seeing is that the case control ratios are like twice as much in the red group. And so you could do like that whole pathway to see if the whole genome-wide association workflow, sorry, throwing out random words, on the red and the blue to see if there's actually an association. Already skipped ahead to this slide, but in real human genetics data, we can take all the SNPs for an individual, cluster them by population, and then after controlling for the difference in allele frequencies that we know exist between populations, now test whether there's an association. Okay. So, very succinctly, this population structure, population stratification is a problem. I've already hit on this one too now, jumping ahead of myself. Right, in clinical settings, this is constantly a problem, not just because of population structure, but because of environmental factors. So if we've got a clinical sample with 3, we can bin them into 3 phenotypes, severe, moderate, and none. But then you do population structure analysis on them and you find out there's actually significant population structure underlying that. This is the same question as the red and the blue subtypes, right? What you would want to do is run your GWAS comparing the phenotypes of these 3 groups within I'm sorry, these pie charts are populations. So like the red group is 75% green population and whatever 30% red orange population, 10% yellow population, so on. So you could test within them, within each color across those phenotypes. Okay, so and that will give you your best chance to discover variants. So go back to the CoLab notebook. And you're going to actually want to open the other one as well. So this PopStruct notebook. And this one won't run on its own. But what we're going to do is take this whole notebook and paste it onto the bottom of this notebook. Oh, you know what? We didn't run all the cells here. Before you do that, got carried away with I got sidetracked on false discovery rate. The QQ plot, right? Not surprisingly, we don't see anything that's super highly associated. Our QQ plot, there's not much that deviates very far from the expected random distribution of the test statistic. Polygenic risk score. This is one of the things that I've sort of cut for time. Something that somebody could do as a project is work with real human data and generate polygenic risk scores. What is that? So many human phenotypes, not too surprisingly, are not single mutation causes phenotype. It's your genotype over many SNPs that all contribute cumulatively to that phenotype, or your susceptibility to a disease, for example. So polygenic risk scores were a way to say, given the phenotype, given the genotype across all 200 in this example, SNPs, what's your relative risk? So polygenic across all the SNPs, what is your risk? And so what this is saying is that in our 0-1 trait binary, like say this is controls on the left and cases on the right, the genotype across all 200 SNPs tells you that you are maybe twice as likely on average, maybe 1.5 times more likely on average. If there was no signature in the genotypes at all, the polygenic risk would be at the same level. Okay, be no increase. And one of the reasons why that's often important in a clinical setting is that you can give an individual a polygenic risk. Okay, so and this happens when you go get genetically tested and you see a genetic counselor to help you interpret it. But like individual number 4, based on its genotype across all 200 SNPs, is 1.69 times more likely to have the trait than not. Okay, and if this is a disease risk, maybe that's appreciable. And in the human genetics world, that means if it's risk for some kind of blood pressure, they put you on a medication, or they wait and see whether you get high blood pressure. That's right. The implications in the human genetics world. And then that just lets you save it if you want to. Okay, so now I've already copied mine. We're going to paste that cell. Let's create a new code cell and paste.
So sorry, just to clarify that polygenic risk score is because they have multiple SNPs that are contributing to the trait.
Yeah, think of it as the odds ratio, but extended to all the SNPs. So where the odds ratio is telling you for a given SNP, do you have increased risk? Polygenic risk is across all your SNPs, what's your elevated risk? Okay, so all I did was take from, just make sure you're following where we're at. I took the PopStruct notebook and copied that cell into my GWAS tutorial notebook. And now I'm going to go back up to the top. I'm going to delete my simulated VCF just so it doesn't mix up which data set I want it to look at. I'm going to delete my old results just so I don't get mixed up. Okay. Everybody with me so far? So we don't need to reinstall the dependency, so you don't have to reload that. But this time, instead of simulating, oh, skipped a step. You need to download the genotype VCF and the phenotype VCF. That phenotypes is not a VCF, but it doesn't matter. I think phenotypes actually downloads TSV. Yeah, it should be. Your file is phenotypes.tsv. So here, this is the first time we're running this one that's going to let us upload a file. And so we're going to pick that genotypes VCF. And you should see a little counter that uploads the genotype data.
Is there a benefit to loading it in code blocks like that versus just dragging it?
Nope. Just the way I put it in there. So the benefit, the reason I picked to do it that way is I can never remember. Well, I do this time just because I've struggled with it so much. But when you do it that way, it's defining the path to that thing at the same time. Anyway, that's why I did it. So if you opened that genotype VCF file, you would see what I'm showing you right now, like 2,000 SNPs-ish. Okay, just going by how many rows there are. And then how many individuals? We've got a bunch. 400 individuals. So this is still tiny data set by human genetic standards, but we're just looking at the SNPs on chromosome 22. Simulated, but sorry. Okay, so we've uploaded it. We don't need to simulate. Whoops. Sorry. You have to run this one so that it knows what the path to that file is. All right, so checking if the VCF was uploaded. If not, it would simulate it, but it was, so it doesn't simulate, but it still specifies the path so that this cell knows where to go get that data. And you can tell it didn't simulate it because remember, first time we ran that cell, it checked there was no VCF there, so it generated that simulated.vcf. This time it just left our data alone, but it's generated now the genotype matrix. Now we're going to load our phenotypes. So that was the other file that you got from Canvas, phenotypes.tsv. And you'll notice that this file has the subpopulation specified. So we're not actually doing, or we're sort of telling it which one we're in the blue circle and which one we're in the red circle from our cartoon, right? So run our logistic regression. Takes it a little bit longer because there's a lot more data. This one is just generating the odds ratio output. So you should see the association results file. So that's got our odds ratios and everything. We're going to now generate a plot. And so far, we've run the same pipeline we did the first time around on this data. So we have not accounted for the population structure in this data set. Because our first time around, there was no population structure. This time, I've already told you there is population structure. But this is also, actually, go back to the Bonferroni correction. It's still, you've got a ton of SNPs there that look like they're significant, right? If we run the QQ plot, everything has jumped off the line. So that should be a red flag. If you're doing this in real life, not this isn't real life, you're doing it in real life. I mean, this is the same thing you would do. But if you see a result like that, that should be the first indicator that there's something wrong, like you're missing something in the data. Because every SNP can't be higher than expected. It's like Wobegon. Anybody know that reference? Anyone? No? Garrison Keillor? No? Man, so old. Lake Wobegon is so it was an NPR show for a long time. But Lake Wobegon is a town, it's a fictional town where all the children are above average. So anyway, all of the p-values are higher than expected here. That means that can't be true. And so the first thing that you would probably want to test, the PRS doesn't really matter because we know there's something going wrong here. The cell at the bottom that we just pasted in is going to rerun the GWAS, accounting for population substructure. So it's going to use that subpop information. And it's basically rerunning the whole pipeline, but in one cell. And so you can see it generated the QQ plot and the Manhattan plot again at the bottom. So now when you correct for population structure, you see basically there's nothing significant. You might argue that maybe there's some signal there in the most associated, but at Bonferroni correction, there's nothing significant. So just a demonstration of what happens if you have confounding variables. And the biggest one is typically population structure of some kind, where there's just an artificial association between allele frequencies and whatever your trait is. Questions? So you have GWAS pipelines now, and there's tons of human genetic data out there that you could do final projects on to do, estimate polygenic risk score.
Did this cell that we just put in, how did it know what correction to use? And then is that a correction we could use every time we have a problem like this? Or do we have to inherently know?
So we're going to talk about that when we get to machine learning. So this one just used the subpopulation definitions that I gave it. And those were simulated. So I defined how much population structure was there. What we'll do, so we're going to do transcriptomes next. And then, so transcriptomes connection here is we're going to map RNA-Seq reads and estimate gene expression data. And now instead of having our one disease trait, we have thousands of traits. Like every gene is its own trait. We can do GWAS on every gene to see whether there's an association between the genetics and the expression. So hopefully we'll get to that by the end of next week, or actually maybe hopefully Tuesday. Then we're going to move to machine learning. And that's where we're going to talk about how do you discover that population structure. We're going to build a classifier, say, given this genotype data, what's the population structure? And so once you've defined that population structure, now you're back to this pipeline. That is a good question. That is the question that you should be asking as a novice bioinformatician. How do I correct for population structure? Okay, so we have 20 minutes. I'm going to start the intro to transcriptomes. So if you go to module 8, there's a new PowerPoint there. Oh, that's an X. So you can't see module 8. Now if you refresh, you should be able to. Sorry about that.
Yeah, you didn't upload the course material.
It says the module is published. Lecture content.
Course materials.
Oh, refresh again. So this is all about setup for, what are we actually doing? What are we going to do on Thursday? What does the data look like? What are the problems or the computational challenges of transcriptome analysis? That's not it. I hope I didn't upload that one. First slide looks like that, I hope. Okay. So the RNA-seq workflow, at least the first few steps, are not that different than the GWAS workflow. You would make sure your reads are good with some kind of like FastP. There's no reason not to use FastP for RNA-seq, just like you used it for the genome reads. Then we're going to map. And then where it diverges is, instead of calling variants, you could call variants, we'll talk about that, but we're going to try to quantify the number of reads that came from a particular coding sequence. So it's really the read depth that we're interested in now, not the variants themselves. So library prep, a little bit different, just in terms of extracting RNA instead of DNA. One of the benefits of doing RNA sequencing beyond just quantifying DNA expression is that it can improve reference genome assemblies. So we talked about, I guess we didn't talk so much about assembly, but one of the problems with lots of genomes is that, like, you think of a genome in terms of chromosomes, but actual genome sequence assemblies are often broken up into contigs, so they're not contiguous. Doing RNA-seq can link them, because if you've got an exon on one contig and an exon on another contig, that tells you that those must be actually connected in the genome. So RNA-seq can give you information about the genome assembly. It also, if you map it to a reference genome, it can improve the annotations, like where are the intron-exon boundaries, measuring differential exon use, so alternative splicing, talk about how that causes a problem as well. You can detect SNVs, like I said, you can use this pipeline. You can to call variants. You can measure allele-specific expression if there are SNVs. And then one of the things that we didn't even really know about until RNA-seq became a thing is there's lots of antisense transcription. So there's a lot of transcription that happens that never gets turned into protein. So all the regulatory RNA, well, not all of them, but many regulatory RNAs were only discovered because of this technology to be able to sequence the RNA. A key note, this is mostly for the people who are actually going to do this. Lots of labs in our department do this. RNA is inherently less stable than DNA, and so when you do quality control, it's measured as something called RNA integrity number. It's oversimplifying it as sort of the ratio between the 28S and the 18S peaks. But what you often see, like this one, RIN10 is like the best. So this profile in the top left, that's really good RNA. And then you get down here to RIN3 and 2, and you've got a whole bunch of peaks. You can think of this as just RNA that is broken into pieces, lots and lots of little pieces. The x-axis here is the size. So you have very specific expected sizes for the 18S and the 28S ribosomal regions. And then as the RNA gets more degraded, you see alternative peaks showing up. Okay. Lots of sequencing centers won't even go, you send off your RNA to get sequenced, or you're doing it here at the Genome Center. If your RIN number is not 8 or better, a lot of times they won't even go forward into library prep. Okay. So this is examples from my lab. And the reason I put them in here is because, again, our department has labs that work on basically everything, lots of non-model systems. Not all RIN numbers are read accurately by the Agilent software, which is the bioanalyzer that does these chips. Beetles, ribosomes don't break apart the same way as mammal ribosomes. And so we had the worst time trying to get somebody, Novogene, to do our RNA-Seq the very first time around. Back in the early days. Again, I'm so old. The mouse ones I highlight because that's what it looks like when you have DNA contamination. We had this extra peak out there past the 28S region, and that turned out to be DNA. So you learn to recognize the patterns. And again, for those of you who are doing this, these are the ones that you will see repeatedly. Doesn't mean you can't go do the RNA-Seq. It's just, you have to know I've got DNA contamination in that. What you could do is treat that with DNase and try to get rid of that without having to redo your whole extraction. That all leads up to this part. The major constituent of any RNA extraction is going to be ribosomal RNAs. And so if you're going to send that off for sequencing, ideally you want to get rid of the ribosomal RNA so you're not wasting all your reads on sequencing the same ribosomal RNA over and over and over again. And so there are multiple ways to do that. This graphic is just basically showing you, right, you grind up your tissue. In that initial pool, you've got some leftover DNA. Mostly you've got processed and unprocessed messenger RNAs, and then lots and lots of ribosomal RNA. So if you just went and did RNA-Seq on that pool, you would get something like this, where that big stack right there, this big stack is you wasted, half your reads just sequencing ribosomal RNA. So ways to get around that, there are kits, like RiboZero is one of the kits that will go in and selectively degrade the ribosomal RNA so that you're not getting that in your library. You can do cDNA capture. So if you've got a well-annotated genome, you can make probes that will anneal to just the genes that you want to survey and pull those down, and then you can send those off for sequencing. And then poly-A selection, which is the most common, at least for eukaryotes. Ribosomal reduction is probably the one that gets used most often in prokaryotes. But in eukaryotes, you just prime off of the poly-A tail. Okay. So that gets us to the end of library prep. Now you get to what are you going to do with your sequencing? You can basically divide RNA-Seq's mapping step into two types. You can map to the reference genome, which is what we did for genome-wide association study, okay? Or you can assemble a de novo transcriptome. So this is where you would take all the reads from your samples and try to assemble those into what the transcripts originally looked like. And because we're dealing with Illumina again, short read assembly is a challenge. Benefits here, if you have a reference genome, it will improve your ability to reconstruct the transcripts because you're lining everything up to a genome, and so you know which exons or which copy of the gene it belongs to. The problem is most species, when it says it excludes the majority of species, most species don't have a good reference genome. And if you try to map the reads to a different strain or a different species, rapidly evolving genes are disproportionately lost from the mapping, right? So if there's positive selection, right, the most interesting genes are the ones you tend to lose. So that creates problems. If you're mapping something, you're doing RNA-Seq on a new species, but mapping it to some other reference genome. The de novo transcriptome gives you gene sequence and expression data for non-model species. That's the upside. but we're going to talk about how assembly is problematic, not always easy to do, and you don't get any information about the genes that are not expressed. We'll talk about that in a second when we talk about assessing the quality of our transcriptome. So you always have this question, are they really absent, like not in the genome, or were they just not expressed? So this is sort of the same slide from genome-wide association studies. Most RNA-seq is still done on an Illumina machine, but long-read technologies like PacBio is increasingly common. When you do RNA-seq on a PacBio, it's typically called ISO-seq because it's giving you all the information about the whole isoform, because you're reading basically the whole transcript in one go, rather than Illumina, which is chopping it up into 150 to 300 base pair pieces and then making you reassemble it. So all of the problems with assembly that we're going to talk about on the next slide and mapping kind of go away with the longer read technologies, but they're still sort of expensive in the sense of you get lower throughput. So we'll talk about why high throughput is important. And the per base error rate is higher, which also increases the cost. It means you've got to do more sequencing in order to correct the base calling errors. But it's coming along, and maybe in a few years, everybody will just be doing long read sequencing for RNA-seq. So this is the schematic of what's happening and why RNA-seq mapping is a little different than genome mapping, because you have to be sort of exon aware. For example, this is our reference genome up at the top, but we're not sampling from the genome, we're sampling from the transcripts. So if exon 1 and exon 2 are together in the transcript, the reads are going to map differently than an isoform where exon 1 and exon 3 are next to each other. And so your mapper, your aligner, has to be able to deal with things like the split read problem. So what this is showing is the first read on our paired end mapped just fine to exon 1, but the second read, it reads right through exon 1 and exon 2. So a minute ago I said one of the benefits of RNA-seq to annotation is that it defines exon-intron boundaries. That's a perfect example for that is perfectly defined exon-intron boundary, because the read goes right through it. The intron is not even there in that read. And then what I've talked about the assembly is that on this read, sort of second from the bottom, exon 1 and exon 3 are present in the same paired end sequence. So you know that if this reference genome wasn't assembled, like those were on different contigs, then exon 1 and exon 3 must be on the same contig because it created one transcript. So these alignments to the exons end up looking like the genome variant analysis, where we've got these pileups, and the sort of added issue with calling SNVs from this data is that you have the potential for allele-specific expression. So if you remember way back when I talked about error calling and quality control, I said in this example where we've got one read that has a different nucleotide in it, do you want to call that a variant or not? When you're doing genome sequencing on an individual, if it's heterozygous, you have a clear expectation for 50-50 read frequency. But when you have RNA-seq data, you can't make that assumption anymore because we know allele-specific expression is a thing, right? You might only ever express mom's copy of a given allele. And so the fact that this is all G's in one C could just be super strong allele bias in the expression. Like you're almost always expressing one allele. And so you have to be a lot more careful about your interpretations when, and in general, it's just dangerous to call variants from RNA-seq data. Can be done, you just have to be cautious with it. So that's probably a good place to stop. So that's mapping to a reference, but what happens if you don't have a reference is the next thing. So we'll talk about, I decided for the sake of time, I've given you the video for how Trinity works. It's a 3-step process. So the link is here, but it's also in Canvas for today's lecture. If you are planning to do this for your projects or whatever, you've got RNA-seq data, knowing how Trinity works is very valuable. But we'll get to that stuff on Thursday and do practice analysis.